Hi, everybody, and thanks for the introduction. My name's Eli, and I'm an MD PhD student at the University of Pennsylvania. Today I'm going to be talking about defining and predicting transdiagnostic categories of neurodegenerative disease. This is work I did with my supervisor, Danny Bassett, and our collaborators, John Trojanowski and Virginia Lee. So I'm just going to briefly outline the talk. First, I'm going to start with some background, get into our results. So we identified clusters of neurodegenerative disease patients by accounting for multiple forms of pathology. Then we characterize those clusters in terms of phenotype and genotype. Then we try and predict disease labels using CSF testing. And by disease labels, I mean both these cluster assignments and like gold standard autopsy diagnoses. So just to give a brief overview of neurodegenerative disease, sort of the gold standard way to study neurodegenerative disease is postmortem autopsy, where you can use specific histopathological stains to quantify pathological protein aggregation. And from these studies, we've been able to associate the presence of specific protein aggregates with sort of clinical syndromes. I'm not going to go into the details of the differences and the symptoms of these syndromes, but basically what we've learned is that amyloid beta and tau aggregation underlie Alzheimer's. There's a collection of tauopathies that are characterized by tau aggregation alone. Parkinson's disease and dementia with Lewy bodies is characterized by alpha-synuclein aggregates, and TDP-43 aggregates underlie ALS and frontotemporal dementia. However, if you take a more comprehensive look at the autopsy data, you see that you can actually find basically all of these proteins in individuals with all of these diseases. So that sort of challenges this model that each disease is characterized by just one or two proteins. So how do we approach heterogeneous diseases like this, especially ones where the ground truth is sort of difficult to obtain until it's far too late? And the important first step is that we use disease models that incorporate all of the relevant features. So in our existing model, we imagine that there's tau and amyloid beta, and if you have too much of both of those, then you have Alzheimer's. If you have a little bit, then you're healthy. But what if we're missing a disease subtype by not including alpha-synuclein, which is found in patients on autopsy with Alzheimer's disease? This might represent, if we add this axis, a new subtype of disease. And without acknowledging this subtype, our diagnostic tests might have limited accuracy. Which brings me to the next point, which is that we should use diagnostic tests that align with our disease models and differentiate patients with respect to the most distinguishing features of disease. So in our project, we're going to try and get at both of those things by first identifying clusters of neurodegenerative disease patients by accounting for many types of regional pathology across alpha-synuclein, tau, amyloid beta, TDP-43, and other features. So we use the Integrated Neurodegenerative Disease Database collected at Penn. This is a database of autopsy cases. Basically, for each of these highlighted regions, the expert neuropathologist quantified the levels of amyloid, alpha-synuclein, tau, TDP-43, senile plaques, angiopathy, other cellular features. This is a very heterogeneous sample, which is good. This is sort of what we wanted to get at. Alzheimer's disease is the most prevalent disease, and this is just primary diagnosis. Patients can have more than one diagnosis in the sample. There's a lot of Lewy body dementia and Parkinson's disease, progressive supranuclear palsy, and then smaller amounts of a whole host of other disorders. And what we're going to do is so for about 16 regions and 9 or 10 scores, it came out to about 152 features. So instead of this three-dimensional axis, we're going to look at 152-dimensional axis of pathology and try and identify clusters of patients in that space. And the way we're going to identify these clusters is by constructing a network in which each patient is a node. And the edge between each patient is defined by the similarity between those two patients across all these 152 pathological features. Then you can use an algorithm. We use modularity maximization to identify communities in that network. And a community is a collection of nodes that is internally dense and externally sparse in terms of its connections. This algorithm has a free parameter that sets the resolution and granularity of the communities that you identify. So we swept through choices of this parameter and picked the value that gave us the most stable estimates of communities, and that gave us 4 clusters of patients. So then we can look at these clusters in terms of the original diagnoses and see how did they sort of separate patients out. And what we find is that these clusters really respect the existing disease definitions. We have one cluster that has a lot of tauopathies, so corticobasal syndrome, Pick's disease, progressive supranuclear palsy. We have another cluster that primarily has Alzheimer's disease, another one with TDP-43 diseases, ALS, and frontotemporal dementia, and then another cluster with synucleinopathies, multiple system atrophy, dementia with Lewy bodies, and Parkinson's. Then you can look at, on the left here, at the sort of representative pathology for patients in each of these clusters, and you find that, you guys see the mouse? Cool. Cluster 1, which is characterized by tauopathies, has the most tau. Cluster 2 has a lot of amyloid and tau. Cluster 3 has a lot of TDP-43, and Cluster 4 has the most synuclein. So this sort of lines up with our expectations. So now this brings us to the next step, which is to characterize these clusters in terms of phenotype and genotype. So the first thing we looked at was the ApoE genotype, which is a well-characterized risk factor for Alzheimer's disease. And as we would expect, we found that cluster 2 had a higher prevalence of ApoE4 allele, which is the risk allele. Cluster 2 had a higher prevalence of that relative to the other clusters. That makes sense because it's primarily composed of Alzheimer's disease. But what happens if we get rid of all the patients who met criteria for Alzheimer's disease from that cluster, and then see if the remaining patients who were categorized in the same cluster based on similarity across pathological features without actually meeting any criteria for Alzheimer's disease? And what we find there is that even without patients with Alzheimer's disease, cluster 2 still has more ApoE4 than at least cluster 1 and cluster 3. So this is pretty interesting because it suggests that perhaps ApoE4 is not a risk factor for Alzheimer's disease per se, as it's currently defined, but histopathological syndrome very similar to Alzheimer's with slightly different boundaries when you account for many forms of pathology. The next thing we did was look at CSF protein testing. This is a commonly used clinical test, and what you expect, so we measure the levels of CSF amyloid beta and CSF tau. In patients with Alzheimer's disease, you classically see low CSF A beta and high tau. And sure enough, that's what we saw. Cluster 2 had lower CSF A beta than everyone else, high phosphorylated and total tau. And again, when we remove patients from cluster 2 that have diagnoses of Alzheimer's disease, cluster 2 then still has lower median CSF A beta than other clusters. Again, this sort of suggests that the current histological definitions of Alzheimer's don't necessarily separate patients clinically in the most accurate way. So using this information, we decided to see how accurately can we predict diagnoses just using CSF testing. And the way that we did this was to use generalized linear models, just a simple algorithm to basically distinguish each disease label from all of the other patients. So we said, let's predict Alzheimer's versus everybody else, Parkinson's versus everyone else, cluster 1 versus everyone else. And we evaluated the performance of these algorithms out of sample using repeated k-fold cross-validation. And what we get is a very high area under the curve for predicting Alzheimer's and cluster 2 at 0.94 and 0.91 respectively. Then, let's see. So cluster 1, which contains tauopathies, had an area under the curve of 0.86. We weren't able to train a model to predict any of the individual diseases that make up this cluster because the sample sizes were too small. But by lumping these diseases together based on pathological similarity, we're now able to distinguish them with high accuracy from a heterogeneous population. And then cluster 3, we were able to predict that with an area under the curve of 0.83 compared to one of the diseases that's frequently a part of cluster 3, frontotemporal dementia, and we were only able to predict that with an area under the curve of 0.75. So overall, our conclusions are that accounting for co-pathology identifies patients who are clinically, genetically, and pathologically similar to Alzheimer's disease, yet they don't meet the low-dimensional histopathological criteria. And then CSF testing can resolve these clusters a little bit better than existing diagnoses. So future directions for this work, I would be really excited to get into some quantitative pathology data. Our pathology scores were semi-quantitative for each region on a scale of 1 to 5, basically. But if you have continuous data, you can apply some cool dimensionality reduction techniques. And along the lines of what Andre Marcan was talking about in his talk on heterogeneity and psychiatric illness, dimensional models can be really powerful. And it allows everyone to have a sort of a little bit of several dimensions. If we could use whole genome sequencing data to predict cluster membership, that would provide very early estimates of disease risk, as early as birth, potentially, I don't know if people would want to know that information. And then we could use these statistical models potentially to generate probabilistic estimates of cluster membership in living individuals, and perhaps that could be compared with treatment response data. So thank you very much. I'd like to just acknowledge my mentor, Danny, our collaborators, John Virginia and John Robinson, and most importantly, patients and families for making this study possible. Thank you.